right, welcome back. Today we're going to be talking a little bit more about mitosis. So far you guys should have gone over the stages of interphase and talked about what occurs during interphase, which remember interphase is the period of time prior to the cell dividing. So it's basically the cell living its life and in some ways preparing for division to occur. We're going to be skipping around just a little bit, so we're going to skip to 2.5.4, which asks us to describe the events that occur in the 4 phases of mitosis. Now I want to just emphasize a couple things. Mitosis simply is the division of the nucleus, and so a lot of people describe mitosis as the complete division of the 2 cells, but actually what it is the forming of 2 identical nuclei. So technically at the end of mitosis you still have 1 cell, but it has 2 identical nuclei within it. This process is then followed by cytokinesis, which cytokinesis is the actual physical division of the cell of the cytoplasm. So please keep that straight in your definition, that we don't have 2 cells until cytokinesis has occurred. Now, right now I'm not going to go through each of the phases. You are going to have your assignment to work on that, and like the directions here say, describe in detail the events of each phase of mitosis and include a labeled drawing of each phase. So you'll have a little time to work on that today. So we are going to move on to the next assessment statement, 2.5.5, which asks us to explain how mitosis produces 2 genetically identical nuclei. So the process of mitosis is going to be kind of the details, the mechanics of how this is accomplished. But what this is asking more about is what parts of the process ensure that these nuclei are in fact identical. So we've got a couple phases here to discuss. The first is interphase, and specifically we're going to be talking about S phase. And S phase, remember, is short for synthesis phase. And in this period of time, each chromosome is copied exactly. And during this time, there are several checks and balances, different enzymes involved in making sure that the DNA is copied exactly. And so that is one part of the process where an identical nuclei is ensured. And then mitosis itself gives us 2 identical daughter cells. And again, like I said before, it really is just about the nuclei. We don't actually get the 2 cells until after cytokinesis. But mitosis ensures that each cell will obtain a copy of each of the chromosomes. And so there's several steps within the different phases of mitosis that work to organize this massive amount of DNA. And so these steps are going to help ensure that each new nucleus gets 1 copy of each of the parental chromosomes. Okay, so before we move on to our next assessment statement, I just real quick want to cover some DNA terminology. So what you see here as a whole, this is what we're going to refer to as a chromosome. And so this is what condenses in order to stay organized so that each new cell will get a copy of the chromosomes. Within this, though, we've got a few different pieces. So each side of this chromosome represents what's called a sister chromatid. And the sister chromatids are identical in every respect. Okay, so remember that in S phase, the DNA was all replicated. Here are where the 2 replicates stay organized. So they are going to attach to one another using a protein called a centromere, and they're going to stay attached until it's time for the division to occur. And so the whole reason for this attachment and this condensing and this organization is to help to keep the genetic material straight, because remember, there are so many chromosomes that are being made and moved and used throughout this process. So organization is really important. Okay, so now that we see that mitosis ensures identical daughter cells, we're going to talk about why would we need these identical daughter cells. So basically the purposes of mitosis are stated here in 2.5.6 that asks us to state that growth, embryonic development, tissue repair, and asexual reproduction involve mitosis. So basically, these are the things that cell division can accomplish. So if an organism needs to grow, it's going to need more cells. So cell division occurs. When an embryo is developing, obviously it starts as one complete cell and needs to grow into billions of cells. So the fertilized egg is going to divide to form a multicellular organism. And remember from topic 2.1 that this will also involve differentiation. So though the daughter cells are identical genetically, they may not appear identical physically. It can also be used for tissue repair, so if there's some kind of loss of tissue or damage of tissue, it can be recovered by replacing dead or damaged cells, which again, to get those new cells will require mitosis, division. And then asexual reproduction. So here we're talking about one organism reproducing without a mate. And so this is accomplished by cellular division. So we've got a parent cell. It will divide by mitosis to produce two daughter cells. And remember that in this case, the offspring will be clones of the parent. In some ways, it's a similar concept to binary fission. However, the process is going to follow the steps of mitosis rather than the steps of binary fission. All right, so that's the end of this video. Please take a look at your textbook to get ideas about each of the stages of mitosis. Thanks.